Okay, so triple sugar iron is the last of the metabolic tests necessary for a gram-negative bacilli. So triple sugar iron is specific. Okay. Triple sugar iron is specific for enterics. Enterics, this is a term we need to know as well. Remember, you needed to know coliform for the last one, for the last gram-negative bacilli test. And coliform has a specific definition, as does enterics. So enterics are gram-negative bacilli that are facultative anaerobes. Notice that this is a lot less stringent than coliform, which required a bunch of other things. So enterics is kind of a bigger category, and E. coli would fit into enterics, but it's not going to be one of the organisms used in this lab. Most enterics are found in the lower intestine of vertebrates, very, very common in the lower intestine. They are all facultative anaerobes, so remember what that means, that they prefer an oxygenated environment. They will ferment without. Okay? So triple sugar iron uses a really interesting kind of tube called a slant tube, And so the slant tube is going to be agar that's poured into a tube, and then you just settle it at a slant for it to solidify. So, back in the 70s, people would make Jello this way. You just, pour the hot Jello into whatever container, and if you wanted to make it real fancy, you just put it on its side, and it would just solidify in whatever slant you gave it, and then you could put another color on or whatever. But for the slant tube, the agar is made and poured in the tube, and then the tubes are set at an angle for them to solidify so that we get two areas to the tube. We have one area that's the slant. So this is the flat area here that's indicated by this top blue arrow. So notice that it goes all the way from the top left to the bottom right. That's the slant. That slant area is the aerobic portion of this tube. And frankly, up until this point, everything that we have looked at in terms of trying to grow it, at least on agar, has been on the surface. And we've spoken about how difficult that could be for certain organisms, certainly for obligate anaerobes or microaerophiles. We couldn't possibly grow there at all. And it was difficult for the aerotolerant anaerobe as well. But this tube, the slant tube, gives us two areas of growth. The slant, which is aerobic, but then the butt, which is anaerobic, and no one ever forgets this term, the butt. So the butt is indicated with this blue arrow at the bottom. But notice that the entire thing below the slant is anaerobic. So the slant is actually the smallest portion of this tube. The majority of the tube is not, the agar in the majority of the tube is not in direct contact with oxygen, with the air. Okay, so that's really important to understand. Triple sugar iron is another chemically defined media. So that means it's very specific what it's composed of. And so the triple sugar refers to the three sugars that are contained in the agar. Remember that the agar itself is not digestible. So the three sugars are glucose, lactose, and sucrose. Glucose is in the least amount. It's only 0.1%, whereas lactose and sucrose, though they are 10 times more, are still not a lot. So there's not a ton of sugar in here, but there are three sugars. So glucose is a monosaccharide, one sugar, whereas lactose and sucrose are disaccharides. All organisms prefer glucose. It's simple. It's a simple monosaccharide. It goes directly into glycolysis without having to make any special arrangements, as I like to say, ahead of time. If you start with lactose or sucrose, well, both of those are disaccharides, and the first step would be to digest the link between the two sugars that make up the disaccharide. If we looked at lactose, lactose is glucose and galactose. So only half of it is glucose and sort of ready to go into glycolysis. Galactose has to be altered a little bit before it too could enter glycolysis. Sucrose has the similar problem. It's also a disaccharide, half of which is glucose, the other half of which is fructose. And so it is also not quite ready to drop right into glycolysis the way that glucose is. It's purposely done in this manner, triple sugar iron, to have the least amount of glucose because all organisms prefer glucose. The idea here is to ask what will happen once the glucose runs out. So the sugars are going to be there as a source that can be fermented, a nutrient source that can be fermented. And we remember from our conversation in mannitol salt agar that fermentation would produce acids, and you remember that from lecture as well. And different organisms will produce different end products, and how much acid they produce differs. We know all this already. So remember, if we want to assess whether fermentation has occurred, then I'm looking to see whether acids have been produced. And so for that reason, we have phenol red as part of this mix. So phenol red, and here it's really easy to see in this image of the slant tube, this is the neutral color of phenol red, which is sort of an orangey pink. It's not pink, but it's not orange either. It's sort of orangey pink. So phenol red is there to indicate to us if there's been a pH change. The other thing to eat in here is peptone. Peptone, remember, we had some of that in mannitol salt agar. Peptone is a source of amino acids. The breakdown of amino acids would produce amines, NH3 groups, which are basic, and those basic groups would make the phenol red go a different color. So we remember that as well. So that explains the triple sugars. Now the iron part is the ferrous sulfate. So ferrous sulfate is iron linked to a sulfate. Okay. Iron is notoriously difficult to keep in solution. Iron is a heavy metal. So we have iron, of course. We know that we have that in the heme, which is part of hemoglobin. Okay, hemoglobin is a protein, isn't it? And the heme is kind of a grid that is held within that protein, and in the center of that grid is an iron atom. And it is the iron atom which actually binds to molecular oxygen and also to CO2. Notice what is actually holding the oxygen is the iron. Okay, so the entire hemoglobin is essentially a protein cage to hold the iron in solution, because iron is a heavy metal and will not remain in solution on its own. If I put iron into a tube of pretty much anything, it will fall right to the bottom. It will not stay in solution. So iron has to be in specific forms in order to be soluble, and in this case, it's ferrous sulfate. So I don't see iron filings in this tube here. There's iron in there, though. It's linked to ferrous sulfate, and linked to sulfate to form ferrous sulfate. So it will be there for a specific reason, which we'll describe in just a moment. So the procedure for the triple sugar iron is sort of interesting because we're going to have 4 organisms, which I'll describe in just a moment. 4 organisms, and for each organism, 2 procedures have to be done. We would have one tube, okay, one tube, and this is just showing the 2 procedures that are going to happen. The first thing that you would do is take a sterile loop, and you would, of course, flame your tube of the organism, insert the loop, remove your sample, reflame that organism tube, put the lid back on, put it in the rack, fine. Now you would flame the triple sugar iron slant tube, and you would put the loop in at the bottom end of the slant, all the way as far in as you could on the slant. Now remember, the slant is the aerobic portion, and you would just zigzag your way out as this first image indicates. You would then reflame the tube, close the lid, flame the loop. Okay, you're almost done. The next thing that needs to happen is that we are going to stab the butt, and no one ever forgets this term. So with stabbing the butt, we would use a needle. Now, the needle is essentially just like a loop without the little circle on the end. And some of you maybe by mistake picked up a needle during lab at some point when you went back there by the hoods to grab loops. Maybe you grabbed a needle by mistake, and I may have said, oops, you grabbed a needle and not a loop. So a needle is basically just a long piece of metal on the end of the handle. That's all it is. It's just you can think of it as a loop minus the little circle part. So that too would be sterilized. And now that you would take that sterile needle and again take a sample from the organism tube. And now you would stab the butt. You would go straight down into the anaerobic portion of the triple sugar iron tube. This is going to allow us to see the aerobic ability of the organism, whatever happens on the slant, and the anaerobic ability of the organism, that which would occur in the butt. Remember that for the facultative anaerobe, they will ferment only when oxygen is absent, so in the butt. And so here's our way to check anaerobic abilities. So after we would do that, we would incubate at 37 overnight. The 4 organisms that we utilize for the triple sugar iron test are all gram-negative bacilli. Enterobacter aerogenes, we've already spoken of this one. This was the sort of pinky, mucusy looking one from the coliform analysis. Then we had Proteus vulgaris. We've seen that one before as well. Proteus vulgaris was used in the streak plate part 2. Okay. Then we have Pseudomonas aeruginosa. And remember that Pseudomonas aeruginosa, if we held it on the side, held the agar plate on the side, you would see sort of a greenish tint. Okay, remember we said that it has a capsule. It too is a gram-negative bacilli, as are the other ones. And then the last one is Shigella flexneri. Shigella flexneri is also a gram-negative bacillus. Remember that the entire reason for doing metabolic tests on bacteria is that you cannot tell them apart through a gram stain. A gram stain gives us some information. Certainly, I could tell that I am not looking at Staphylococcus. If I'm seeing pink rods, okay, I've got that part out of the way, but now I don't know which one of these I'm dealing with. So a gram stain doesn't really tell me a lot. So remember that for each one of these, I would treat it just as I showed in the image of the last slide, that a zigzag pattern would happen on the slant, starting from the lowest portion of the slant and zigzag your way up and out of the tube, and then the butt would be stabbed with the organism. So it's very important to do the same organism in the same tube. And this is, of course, not something that you're going to make a mistake with because you're not doing this at all. But sometimes in labs, it just because there's so many tubes on the table at once, some students by mistake will streak, let's say, EA on the slant and unfortunately stab with PV, and they get really mixed results. But that can't happen to you now, so that's fine. So we would have both an aerobic and anaerobic portion for each one of these 4 organisms. Okay, so this is what would happen after the incubation overnight. So you've seen this image before if you looked at lab exam review part 2. This is the same image that I use in the review module. So looking at these 4 tubes, this is what you're going to get. Now notice how different they all look. They look really different. And if you're looking at EA, notice that there's 2 tubes there. Okay, so one is, the one on the left would be the first one, and if the second one would be as if it went like one more day. So it's important for us to understand that. The first tube on the far left just says C, which is control. So that's our control, and each one of these I'll describe. Okay. So remember that phenol red is our pH indicator, and that if we see yellow, then we know that we are dealing with acidic conditions. And if we're dealing with acidic conditions, then I know that acids have been produced, and that is my telltale sign that this organism can ferment. Okay, that's fine. If I see pink or fuchsia, then I know that what I'm looking at is the release of the amine group, and so you'll just indulge this image I made here myself of the amino acid. Any amino acid, of course, has an amine group over here on the left-hand side, and cutting that off, that's a waste product of the metabolism of proteins, is an amine group. And so that ammonia is basic, and that's where I would see fuchsia or pink. So if I'm looking at any one of these tubes and I see pink, then I know what happened. I know they broke down the peptone. Okay? And the other thing in here, remember, it's triple sugar iron. Okay? So I can see how much yellow, and we'll talk about that in just a moment, and we can certainly see pink. The other thing we can see is black. Black is the iron that will happen if cysteine, a certain amino acid, cysteine is one of the 20 amino acids. If that has been metabolized, it will react with ferrous sulfate, okay, to give us a black precipitate called ferrous sulfide. These are the 4 tubes and their 4 reactions, and we will describe them one by one. The first one is Pseudomonas aeruginosa. Pseudomonas aeruginosa is actually in here as a negative control. Pseudomonas aeruginosa is most certainly a gram-negative bacilli, but it's not just being a gram-negative bacilli that makes you an enteric. The other portion is that you must be a facultative anaerobe. A facultative anaerobe has the ability to ferment when oxygen is absent. And yet the anaerobic portion of this tube, which would be the butt, is down here. If you look carefully at the butt of the Pseudomonas aeruginosa versus the control, there's no color change whatsoever, meaning that nothing happened in the butt. There is no anaerobic ability, and in fact, Pseudomonas aeruginosa is an obligate aerobe. It has consumed the sugars, just like you would have. It consumes the sugars in an aerobic metabolism. Aerobic metabolism utilizes glycolysis, the Krebs cycle, and yes, the electron transport chain, whose final products are water and carbon dioxide, neither one of which is acidic. So once they finished eating those sugars, and they didn't make any acids from the aerobic metabolism of those sugars, what else is there to eat? Well, sure, they have to eat the peptone. And when you break apart the peptone, then you will release an amine, and that will turn phenol red this pink color. So that's what we see. I see no evidence whatsoever from fermentation in this tube. I only see evidence of peptone metabolism. Okay? It's very important to understand here that the sugars are eaten first. All organisms eat sugars first because they're easy, that's why. So the sugars are eaten first. After that, then it turned to peptone. Okay? So we can see by looking at the butt that there is no change whatsoever in the butt. And what that means is that it has no anaerobic abilities. It is an obligate aerobe. Notice how I can figure this out just by doing this slant tube test. Okay, the triple sugar iron gives me this metabolic ability on the other side. The next one is Shigella flexneri. Shigella flexneri is a tube over here on the right. Now, if we look at this carefully, we can see a very big difference between the control and Shigella flexneri. Let's look first at the slant. The slant is pink. Okay, the slant is pink. It's actually, this tube, it looks a lot like raspberry lemonade. It's so pretty. So this pink lets me know that some peptone metabolism has occurred here on the slant, but the butt is yellow. So that indicates fermentation. Now I want us to recall something that I said at the beginning of this lab, and that is that the slant is actually just a small portion of this tube. Only a small portion of this agar is actually in contact with air just below the slant, just below, like 1 millimeter below, it's already anaerobic, okay? So this organism is able to ferment. I can tell because I see yellow down at the butt. But the thing is that this organism didn't make that much acid because already up here at the slant, especially starting up here where the slant is thinnest and moving down to where it gets thicker, I see pink. It ran out of fermentable sugar and started on the peptone. It did not actually make that much acid because already the color change has moved into the basic look of the pink. What this indicates is that Shigella flexneri is able to ferment glucose, but that's it. The glucose, remember, is in the least amount. So if the glucose is in the least amount, it certainly fermented it, that's fine, and then it quickly ran out of glucose. It ate the other sugars next. Sugars are always first, but it couldn't ferment them. So it just ate them aerobically. Okay, that doesn't produce acids. And then once the sugars were done, well, it had to turn to peptone. And so we see the pink indicating the peptone metabolism. So this is acid at the bottom, but not that much, otherwise it would be all the way to the top. Okay, so that's what I see is the ability to ferment glucose only because the butt is yellow, but the slant is not. The slant is pink. Let's look at Enterobacter aerogenes. I have 2 tubes of Enterobacter aerogenes shown here next to the control. And they indicate the first tube of Enterobacter aerogenes would be after day 1, and the next one would probably be, it really may be like 6 or 8 hours later. It wouldn't have to be a whole day later, but you can see that there's already a difference there. But the biggest difference you see between EA and the other tubes that we've looked at thus far is this big gap at the bottom. This big gap at the bottom indicates that gases are produced. That's unique compared to what we saw for Pseudomonas aeruginosa, which didn't produce any gases, didn't ferment whatsoever, and Shigella flexneri, which, yes, fermented, but I didn't see any gas. Okay? So we see gas production here. The other thing we see, let's look at the first tube of Enterobacter aerogenes, it's yellow all the way to the top, all the way to the top. So that means a ton of acids have been produced. This indicates that all 3 sugars were fermented, not just the glucose, all 3 sugars, a ton of acid is produced. The phenol red goes yellow, and that's what I see, is that lots of acids are produced as gas is produced as well. If we let this go 1 more day, it would run out of sugars. What is there to eat now? Peptone, that's right. The breakdown of peptone would release the amine and I would start to see fuchsia. But notice, so this second tube of EA, we do see some pink, but only it starts up at the top where the slant is thinnest, okay, where the agar is thinnest, and it starts working its way downward, okay, just like this. You can already envision what is going to happen in 1 more day. It's out of sugars, there's only peptone left, and so as the peptones would continue to get broken down, we would continue to make more and more amines, and those would turn this agar more and more pink. Okay, so you can see that the triple sugar iron test is really time sensitive, because if I wait too long, I'm going to mistake EA, okay, for SF, which was the one before, which was pink further down. However, the gas is unmistakable. Okay, so then one is Proteus vulgaris. Proteus vulgaris, let's look first at the slant. Notice how it's yellow all the way to the top. Yellow all the way to the top indicates that all three sugars are fermented. So glucose, lactose, and sucrose, all three are fermented. But much more dramatic than that is the solid black butt that you see down here. That black butt occurs because of the ferrous sulfate that is in this triple sugar iron. The amino acid cysteine is part of the peptones. Peptones are pieces of proteins, and all 20 amino acids would be present in that peptone mix, including the amino acid cysteine. This is what it looks like. So here's your amine group on cysteine, but cysteine, the R group, the variant group, has a sulfhydryl group right here. If the organism is able to clip the sulfhydryl group off of cysteine, that sulfhydryl will react with the ferrous sulfate to create ferrous sulfide that will fall out of solution and form a black precipitate, which you see in the butt. It also produces H2SO4, which is also acidic, so that's just going to add to my yellow that I see in the slant. So this is really unique. It was the only one that could do it. Okay, this was the only organism that was able to metabolize cysteine. Okay, this reaction here is pretty specific. I usually have it as an extra credit for you to explain what happened in the butt. So the simple answer is it indicates cysteine metabolism. Fine. But how did that actually turn black? Okay, that's the story here. That release of the sulfhydryl from cysteine metabolism, that sulfhydryl reacts with the ferrous sulfate to produce the ferrous sulfide. That's the precipitate. That's what we're seeing. Okay, so look at this mix of triple sugar iron and notice that this is chemically defined. Just like we saw with mannitol salt agar, each component of this media is there for a specific reason. A specific reason. Familiarize yourself with what is in there and what the roles are. Okay, so our recap on triple sugar iron. We utilize a slant tube, which gives us an aerobic portion called the slant and the anaerobic portion called the butt. And the butt is the majority of this tube. Most of it, it's anaerobic. We streak in a zigzag pattern on the slant and we stab the butt. Okay. If we see yellow, that indicates fermentation. The sugars in triple sugar iron are the fermentable source. If they can ferment, they will produce acids, and I will see that because the phenol red lets me know, okay, that acids have been produced by going yellow. If they're out of sugars, they need to turn to peptone, and peptone will, metabolism will release an amine. That amine is basic, and phenol red will go pink or fuchsia above pH 8.4. So if I can look at it from across the room, then I know that this is differential media. Okay, and we can't forget that triple sugar iron also indicates that we have an iron in there, but it's not iron. So don't say Fe to me, because that's not really right, is it? If it was just iron, then I would see, black iron filings at the bottom of this tube. But instead, it's ferrous sulfate. It's in the mix. It's there as a salt, but it will react, okay, and fall out of solution if cysteine metabolism has occurred. So please familiarize yourself with all of those parts.